Hi all, welcome back to Microbiology Lectures, and today we're going to talk bacteriophages, or what sometimes is just referred to as the phages. So the phages themselves have three general kind of relationships or cycles. I don't want to call them life cycles. Remember, phages aren't alive, but replication cycles is kind of a good way to think of this. The first one is the lytic cycle, and so if we look at the lytic cycle, bacteriophage will inject its nucleic acid into a bacteria. That genetic material will hijack, so the proteins and the genetic material that's associated with it will hijack the replication mechanisms of the host. Okay, so it'll hijack the ribosome, the use of the ribosome, the use of the polymerase, whatever it might need in order to copy its genetic material. If it's DNA, it's going to need to make RNA strands of that. If it's RNA, it'll need to make a complementary strand of that, and then translate that into proteins. And so the lytic cycle is normally what we call the virulent or the death cycle. And so what that means is material gets injected in, it gets read, it gets transcribed, it gets translated, it starts building the proteins that are needed for replication to occur, and then it lyses the cell. The cell dies, and the viruses come out identical copies of their original virus. Okay. The other two versions can be a little bit different. So you can have the temperate phages. These phages can do either cycle. Okay. And so the species that belong to temperate phages can do either cycle. They could just go right into the lytic cycle, or they can go into the integration cycle, which we call the lysogenic cycle. So they'd inject their genetic material. That genetic material, linear or circular, can get incorporated in the genome. When it does so, then it's permanently in that genome, or at least unless the bacteria has a defense against it, which we'll talk about CRISPR-Cas9 and some of the other defenses where it can remove that material. But if it does not remove that material, it gets incorporated, and then every time the bacteria goes through binary division, splits, divides, and makes a copy of that genetic material. And so that is a great way for viruses to replicate without destroying the host cell. Then something can trigger this lysogenic cycle to move into the lytic cycle and rupture the cell or start the translation process and build new viruses and rupture the cell. So the temperate phages can move from the lytic cycle, or from the lysogenic cycle to the lytic cycle, or they could just be lytic. So just keep that in mind. The filamentous phages are a little bit different. They're purely integrated. So what happens is, same kind of process, genetic material from a filamentous phage is injected into a host. It gets incorporated in the host's genome, copied, replicated, etc. But it's incorporated to the point where it's being replicated, and the proteins needed to make that virus are being replicated without the need for, basically the reassembly happens outside, or while the virus is leaving that host. So what'll happen is you get these pore-like structures that'll form. The genetic material will make its way out of the host cell, and when it is coming out of the host cell, in the case of like this example here, which is similar to tobacco mosaic virus, which is an alpha helix, while it's moving out, those alpha helical proteins will start capsating or go over the genetic material and then the new virus will be formed after it leaves the host completely. But because it's integrated, this can happen over and over and over again, and the host cell just keeps making new viral units without that host being destroyed. The host, it still maintains its cell membrane and its normal functions, can still go through binary division, it can still consume material and keep bringing in energy, so it has enough energy to make these proteins to keep making those viruses. So really there are two general stages for phage reproduction. You can either have a productivity or productive stage, which we normally call the replicative stage or the lytic stage, where new particles are being produced. Okay? Or you can have the latent stage where that viral genome is just sitting inside the genome of the host and waiting for conditions to be right. Sometimes those conditions means there's enough energy for the host to then produce enough proteins and things like that to make new viral units. Other times it's the conditions of the host is worsening, and so it's time to get out of that host, lyse its cells, move on to a different host. So there are different triggers for moving out of the latent state. Okay, so let's look at lytic phages in a little more detail. Okay, so like a T4 phage, which will often infect E. coli. It's a classic model to kind of follow, and we know a lot about the process of infection in T4 phages. So we're just going to kind of go through this now. Different phages will have different little tweaks to kind of the 5-step process, but the T4 phage, that's what we're going to cover right now. So the attachment would occur. So these phages will come in, attach to the cell wall, sometimes the capsule, but they're attaching to the receptors that are found on that cell wall or that capsule. That allows for them to take hold. When they take hold, they will then drill a hole through the cell wall. A lot of times this is that genome entering is coupled, or the way that this works is the excretion of lysosomes, which will break the linkages between the peptoglycan layers and allow for a pore or a hole to make it through the cell wall, and then they can inject the genetic material in. So you have attachment where the phage are going to have receptors, and those receptors often are specific to a group of species. Sometimes it's a single species, and there's viruses that can only affect a single species. Other times it's a little more general, and they could maybe affect all of one genera or multiple genera. It just depends on the virus. But they will get established by attachment. Then the genome enters via typically some type of enzyme degrading of the cell wall. So lysosomes or other enzymes. There are other enzymes that can degrade it. But nonetheless, those enzymes are made by the genome of viruses. So they hold on to those enzymes because the cell itself, when the virus is replicated, that cell made the lysosomes that is carried by the capsid of the virus. And so remember, viruses can't make their own proteins. So everything that they need to continue the replication, to destroy the cell wall, to start the process of replication, is made by the host cell. Okay, once it's in, then that genome can be synthesized, and so that genetic material will take over DNA polymerase, or if it's a DNA strand that's being injected, it's going to be transcribed into RNA and then translated into proteins. If it's an RNA, then a little less step sometimes is needed, but nonetheless the synthesis occurs, the proteins are being developed, the amino acid chains are being linked together, and you're starting to build the protein structure that's needed for the capsid in the tail. Once that synthesis has occurred and is done, then the assembly process can take place. Okay? And the interesting thing about a lot of the viruses we know of, the assembly is spontaneous. And so the capsid head will often fit to the tail spontaneously. So the amino acids are linked, peptide bond linked together, and all that information is carried within the virus genome on how that comes back together. Once that occurs, then the virus can then lyse the cell of the bacteria. Often that is associated with the same lysosomes it uses to get in, it uses to get out. Okay? Some viruses don't necessarily need that, and they can just rupture the cell because enough material is being built in the cell that it causes the cell membrane to rupture. But we know of some phages that we know approximately how many viral units a single cell, like a single E. coli, can hold. And for like T4, we know its burst size is about 200 units. So about 200 viruses per E. coli cell, and then it'll rupture. Lysogenic infections are a little bit different. Okay, so again, these are stages at which there's a latent period. Okay, so the phage is going to go through the first attachment and insertion of genetic material, but then the genetic material, instead of being copied and translated, so transcribed and translated, instead it's incorporated in the genetic material of the host cell. And normally what happens is there's a repressor that's attached to that phage DNA, and that repressor doesn't allow for the transcription translation process to take place right away. So it gets integrated into the host DNA. When that happens, we call it a prophage. When the prophage is copied, so when that bacterial cell is carrying it and it's copied, okay, those bacterial cells are often called lysogens, or lysogenic bacteria. And so that indicates indeed they're carrying a virus that can go from the lysogenic stage to the lytic stage if conditions are right. Okay? So most of the time, those genetic material from the phage is silent. Okay? It's not being copied. Now, again, we talked about the filamentous viruses. That's being copied, and it can be copied continuously. Okay, so this is a little different. We're talking about viruses that are in the lysogenic stage, but in a latent period. Okay, so they're going to move to the lytic stage at some point in time, but when they're in that latent period, their genes are silent and there's no protein synthesis. Okay, so here's just an example of that. So a bacteriophage comes in, attaches, injects its genetic material. The genetic material can go straight to a lytic infection where it's being copied and then translated into proteins. Proteins form, and the cell's lysed. Okay. Or that genetic material can be incorporated into the DNA of the host, and with it gets silenced based on repressor proteins typically, and then it just lays dormant, and the cell divides and continuously divides over and over and over again until the repressor proteins are removed. When the repressor proteins are removed, it bounces back into the lytic infection and starts the process of replication, so transcription and translation, and builds proteins and then lyses the cell. We know from previous research with certain species of viruses that this can happen, this movement from the lysogenic stage, okay, or the latent stage to the lytic infection can happen about once every 10,000 divisions of a lysogen. Okay? Now, this is species determined. Okay? It's determined by how quickly the host replicates. Okay, so some hosts that replicate, like E. coli, every 20 minutes, that's probably a 10,000 division. But for things that might take a little bit longer to replicate, maybe it's not 10,000 divisions. And again, species dependent. Okay. Other things, though, that can trigger this to happen much faster are changes in the host cell's chemistry or in the host cell's environment. Okay. We know for a fact, because we can do this in labs, if you zap the host cell with UV, that UV light can damage the repressor, and then it will push that virus into the lytic infection cycle. But we know from environmental factors that we can also mimic, or that might mimic, natural occurring environmental factors like changes in temperature. Temperature can drive this process. Changes in available nutrients can drive this process. Changes in pH can drive this process. So there are lots of ways at which that repressor can be removed or destroyed. When the repressor is removed or destroyed, it starts that infection cycle, or that lytic infection cycle, and that really allows for that virus to get out of a poor situation. That host is probably being damaged, that host is not doing well, and that virus can make copies of itself and get out and affect a different host. Okay, so there are some things that we typically say are the consequences of lysogenic infection or lysogenic stages. So for one thing, the lysogen itself, if a bacteria is considered a lysogen, so it has genetic material that's been incorporated in their genome from a certain species of virus, it cannot be affected by that same phage. Okay, so it can't have that same genetic material be incorporated again. Okay, so we often will consider this, it's immune or not able to get a super infection. Okay? And so when the other thing that can happen with that is the repressor itself can be maintained on the prophage for a very long period of time, to the point where it's possible, based on some evidence, suggests that the repressor is on that latent genetic material to the point where mutations within the genome of the host has mutated the viral DNA, so the viral DNA is no longer expressible. Okay? And so sometimes we would consider this genetic material that shows up in an organism that's viral, okay, but doesn't have the ability to produce that virus anymore. It's just an indicator of a previous viral infection, right? And so it's just latent DNA that now sits there, and sometimes we would consider it junk DNA because it just sits there and it has really little to no function. Now, when we look at that junk DNA or that viral DNA, we can look at something like, let's say we look at the human genome and we look at all the genetic or the percent of genetic material that we have, and we know that protein coding genes, there's very little of protein coding genes. Okay, we have structural DNA, we have some regulatory DNA which turns on and off different, protein coding, but then you have all this other DNA. Sometimes it's introns that are between genes, or introns within a gene. Okay, sometimes there's these short random repeating units and long repeating units, which is making up a lot of our genetic material. But you have these fossil virus DNAs, okay, RNA and DNA units that are represented in the human genome. Okay? And we think this is evidence of prior or previous infections from viruses that became incorporated in our genetic material, but are dormant and cannot, the viruses can't be replicated any longer because mutations have happened, or the repressor is so integrated that it can't be removed, that kind of stuff. Okay. Now sometimes, though, what can happen is you can get what we call a lysogenic conversion, and that's where something that might have occurred in the prophage DNA can then be carried, or the organism, the bacteria, could then utilize that. Okay, and so sometimes we see things like toxins that are encoded by phage genes are carried, and the phage can then move that toxin either to a new host, or now that host has a new toxin that it can produce, and so a new maybe a new protein strand that it can produce, or something like that. So you can get this transfer, this horizontal transfer of genetic material when we look at viral DNA. And so the viruses are interesting from that perspective. They can transfer material, and then the host can pick up that material and potentially use that material later on to make some of the proteins that were carried by the phage for the production of the phage. Okay. So with that, we will talk about how our immune system and other organisms' immune systems fight off things like phages, or things like bacteria or protozoans next time.